disinfectant. You know, well, in the old days, if we had cuts, we would go to the school nurse, and the school nurse would take a swab of alcohol, neat alcohol, put it on the wound, on your cut and your scrape on your knee, and it would sting. Why? Because it's not only sterilizing, it's cleaning all the microbes, it's killing your cells. So it's really, really belt and braces approach. But it is wonderful, and it's what they use to sterilize surfaces in surgery. Some of our disinfectants at home have got alcohol in them. Some of them don't. They have different types of detergents. So we used alcohol gel to wipe off more bacteria, and then we tested again on the agar. So we did 3 tests to test whether we had microbes in our hands and whether we could get rid of them with different things. Then we did another. We had 2 more plates. And the other 2 plates were to see what type of microorganisms are in our surroundings. And I had little cotton buds, yeah? And I said, choose some surface, hair, glasses, shoe, equipment, microscope, door handle. I said, try the laptop or the mouse or the keyboard, because people are typing on that all the time and we've got microbes on our hands, so I told you. So then we streaked the surface of these plates, yeah? Take them all out. And then we had one last really difficult experiment for you to do, which was the microbial sensitivity assay, where I had given you 2 plates that look like that. Carmen?
Yeah.
2 plates that look like that.
Thank you.
One of them, the blue one, that one there, I had previously streaked with... Do you need something, darling? You can't see?
No, I'll say it after.
Okay. So, one of them had previously been streaked with a thin layer of E. coli, the gut bacterium, okay? So this is that bacterium. And then the yellow plate, I had streaked with a thin layer of yeast, brewer's yeast, Saccharomyces cerevisiae. Yeah, the 2 names, I've written them at the top. And then I said, let's do something that people do in real life. This is something that is done in hospitals, that is done in universities, that we do in our research institute when we're trying to do investigative science. So we do this assay all the time. And I asked you, a whole bunch of 10 and 11-year-olds to do it. I can't believe that you did such a good job. So then we divided them up in a place like that, yes? And we numbered them, just like in the picture. And then with incredible skill, you picked up a little tiny circle of paper that had previously been soaked in a... I won't ask you for a sample because we've already got some. Otherwise I would ask. So we put circles that have been soaked in different solutions. Water, do you remember water as a control? Everybody always has to have a control. Science demands that we do controlled experiments. Vinegar, I thought vinegar would be something that's really acid. It's strong. It might inhibit or stop the growth of some bacteria more than the others. Then 7-Up. I thought, it's what's in fizzy drinks? Lots of sugar. It's got carbonated water. It's got phosphoric acid, so it's actually quite acid. So in terms of vinegar, it's also quite acid, which is why we like drinking it. Then I said, let's go for the real acid test. Let's go for washing up liquid and toilet cleaner. I thought, and you chose to mix the 2 things together. So we did some chemistry here and mixed 2 different types of detergents together and plonked them on a dish, and then we did the little surface passing. Then we used mouthwash. Mouthwash is what we use. ¿Cómo se llama mouthwash en castellano? Enjuague.
Colutorio.
Never remember that one. So mouthwash. Mouthwash is it's supposed to get rid of bacteria from your mouth after brushing your teeth, just to make sure you don't get tooth decay. And I had some at home, so I brought some. And then we checked with antibiotic, which is what I'd said that would probably kill bacteria. But we needed to test that, because I can say anything. You have to believe me. You don't have to believe me, you have to see the result. Then we used a disinfectant that is alcohol-based disinfectant, which is going to be a bit like our gel, but slightly different. I mean, more. They use that in hospitals. And then we went for Mum's favorite, the one that gets put in toilets and everything, bleach. Yeah, lejía. It's really stinky. I I looked in the bottle when I got home, and guess what it said? Kills 100% of microorganisms. So this time, the producers of, I think it's Sainsbury's, have said, nobody's going to sue me, I'm telling the truth. Bleach will kill every microbe. So therefore, it should kill all our microbes. So then, we did the test, and then I incubated them, and I've got the results back for you. And now is part 2, which is you've done all the hard work, and this is exactly what happens in laboratories worldwide. You do an experiment, you have to do all the hard work for it, you then have to wait, usually for something to happen, time, microbial growth, and now we come to do the interpretation, which is this important part of the experiment, of the experimental design, which is we have to interpret our results. So what I'm going to ask you now is,